Sure. So this is Daniel, and Daniel is actually a Region 6 alumnus, and so that's over by the Charlotte area. He's currently a student at North Carolina State University and is a Park Scholar, studying chemical and biomolecular engineering with a minor in biotechnology. He works under the guidance of Assistant Professor of Chemical and Biomolecular Engineering Nathan Crook, Emeritus Professor of Biology Robert Crossfield, and Mathematics Lecturer Elizabeth Brown, and was recently awarded the Goldwater Scholarship. His research involves working with probiotic yeast, and he's interested in using the yeast to create beta carotene for use in the prevention of vitamin A deficiency. He's currently planning to pursue a PhD in chemical engineering and is thinking it will likely be in synthetic biology, specifically around disease treatment. And so with that, I'm going to stop sharing my screen and pass the mic over to Daniel. Welcome, Daniel.
Thank you so much for that introduction, and hi everyone. Thank you so much for having me. As Teresa said, my name is Daniel, and I'm an NC Science and Engineering Fair alumni from Charlotte originally. I had the opportunity to attend the International Science and Engineering Fair 2 times as part of this program, and it was a really impactful experience for me. So I hope that one day you guys are also able to have that experience. I am now at NC State University, as Teresa said, studying chemical and biomolecular engineering, and I'm really enjoying it there. I encourage all of you to apply. And today, I would like to tell you about my worst research enemy, the biggest obstacle to me in my undergraduate research. This is the challenge that time and time again has disrupted my schedule, wasted my expensive reagents, and laid my experiments to waste. My research nemesis is this. This object that you see in this picture is my worst research enemy, something that my colleagues and I disdainfully call the ledge. The ledge is this little bit right here in the circle that sticks off of the glass box. The box is called an electroporation cuvette, and it's a tool used to get DNA inside of bacterial cells in a process called electroporation. The way it works is you dump your cells into this box, this cuvette, you mix in a little bit of DNA, and then you electrocute the cells. That's right, I hate to break it to you, but bioengineering can sometimes be more accurately described as microorganism torture. Electrocuting the cells forces microscopic pores on their surface to open up, allowing the DNA in. It's a pretty cool technique, and it's not supposed to be too difficult, and yet I failed at it over and over and over again my first year of undergraduate research. And this ledge is to blame. See, when you put the cuvette into the electroporation machine, you have to do so in a particular orientation. The ledge must be facing out. Do you like my PowerPoint graphics, by the way? That way it sits properly within the metal housing and the current is delivered directly to the bacteria. Everything's good, DNA gets in, everything is going according to plan. However, if you're like me, you sometimes forget this orientation and you stick the cuvette in like this. You can already tell right off the bat that this isn't going to go well. When you try to electroporate your bacteria with the cuvette in this orientation, a couple of bad things happen. First, the machine makes a very alarming, high-pitched, high-volume beeping noise, causing everyone in the lab to turn their head and look at you. After that, it displays the word arc, meaning that the electricity has kind of arced around the bacteria, missed it completely, and your transformation has failed. Worst of all, you have to sheepishly tell your colleagues that once again, you have failed your DNA transformation, and it's all because of the ledge. Now, I know what you might be thinking. Dang, this guy is pretty sloppy, making the same mistake over and over again. And maybe there's some truth to those accusations, but I promise you that in general, I'm not a sloppy scientist. I'm careful, precise, and intentional with everything I do in lab. In fact, when I first joined my lab, I got a reputation for writing everything down. I would follow the grad students around with a notebook and a piece of pencil. I would just write down every word that came out of their mouth because they didn't want to make a mistake. And yet somehow I managed to always screw up that ledge. Why? I've spent a lot of time thinking about this, and I think I've come up with a reason. I was trying to be perfect, to avoid making a mistake. Specifically, I wanted to be an expert when I was still a beginner. I wanted to feel like a capable scientist who knew what he was doing and didn't have to ask questions. And so when I put the cuvette into the machine, I never stopped to confirm with my colleagues whether I was doing it correctly. But because of that perfectionist attitude, I ended up making the same mistake over and over again. That's when I was introduced to a concept that changed the way I think about research and eventually changed the way I approach life in general. It's called the beginner's mindset. A beginner's mindset simply refers to having the open and accepting mind of a beginner. Having a beginner's mindset is about having the humility to recognize one's own limitations. It's about asking questions and being open to new possibilities. And if you're a perfectionist like me, it's about giving yourself permission to mess up. I want to point out right off the bat that despite its name, a beginner's mindset isn't just for beginners to be discarded after picking up more expertise. A beginner's mindset is something you can have even when you're an expert. Many accomplished scientists will tell you that when they're tackling a new problem, one of the first things they do is try to forget about their preconceived notions and experience in order to just let their creativity flow. All of this to say, you can adopt a beginner's mindset in any area of your life, research or otherwise, regardless of your level of experience and expertise in that area. And doing so can allow you to become more creative and resilient in the face of failure. After learning about the beginner's mindset, I started making a conscious effort to adopt it in research. For me, that meant asking all the questions. Can you remind me how to do this technique? Why do we do the steps in this order? And most importantly, where does the ledge go again? When I made a mistake, I allowed myself to acknowledge it and move forward without beating myself up. And with time, research started to feel different. Now I realize that I can use a beginner's mindset in so many areas of my life, not just in research. Now, they say that one of the most important parts of giving a presentation like this is knowing your audience. And given that y'all are a crowd of incredibly high achieving high school researchers, and that I have just made a very bold claim about the effectiveness of a beginner's mindset, I wonder if you're all thinking the same thing. Show me the data. I wish I could show you a graph that proved that the beginner's mindset made me a more effective scientist. But not only do I not have the data for this, I'm not sure if it's true. I don't think a beginner's mindset made me a more effective scientist exactly. My experiments probably still fail at exactly the same rate as before. But adopting that beginner's mindset has allowed me to meet challenges with enthusiasm and flexibility, and to bounce back from failure with optimism. However, since this is a science fair, I would like to share with you some cool experimental results from what I've accomplished so far in research at NC State in order to encourage you to consider continuing to pursue research once you get to college. As many of you already know, bacteria are incredibly useful for producing a variety of important molecules like antibiotics and cancer medications. One way to improve bacterial production of a small molecule is to use a technique called directed evolution, which is depicted in this figure you see. In directed evolution, we introduce a bunch of random mutations into the DNA of our population of bacteria. And then we check out the phenotypes, the results of these mutations. We then select for the variants that show improved function. Over many cycles, we can produce microorganisms with drastically improved production of our target molecule. However, this technique is not without its limitations. First of all, conventional directed evolution techniques require a lot of pushing DNA in and pulling it right back out of bacteria, so you can only work with relatively small pathways in terms of DNA length. Additionally, producing all these random mutations in cells means that mutations are going to accumulate throughout the whole genome, not just in the genes you're trying to evolve. At NC State, I'm part of a team looking to get around these limitations by using this guy, a P1 bacteriophage, which is a virus that infects bacteria like E. coli. Our method for directed evolution, called inducible directed evolution, or IDE, works like this. By placing special packaging sites around our gene or pathway of interest, we can actually trick the P1 bacteriophage into packaging our mutated DNA instead of its own. So it does what any good virus does. It picks up that piece of DNA that we've introduced mutations to, it puts it inside its capsule, it busts out of the cell, and then it goes on to infect new, fresh, unmutated cells. Since only our gene or pathway of interest is transmitted, we don't accumulate those hitchhiker mutations in the rest of the genome. Plus, P1 can carry huge chunks of DNA. We've used it to successfully transmit pathways as large as 36 kilobase pairs, but we could hypothetically evolve a pathway 87 kilobase pairs long. For context, that's enough to carry more than 60 copies of the human insulin gene. We've used this technique in a lot of cool ways, including making fluorescent cells glow brighter. This is GFP fluorescence that you're seeing in this picture, and we've engineered cells to glow brighter than they would otherwise. In my opinion, the coolest thing we've done with inducible directed evolution is to make E. coli grow on tagatose, which is an artificial sweetener. Normally, E. coli doesn't like to eat tagatose, but after a few rounds of IDE, we can change its mind. This black trajectory you see on the bottom of this graph is wild type E. coli, unevolved. You can see it doesn't like to grow on tagatose very much. It doesn't increase in optical density very much over time. However, after a few rounds of IDE, you see that we get these other colors of trajectories, the blue and green and gray. Not only do these E. coli strains grow faster on this artificial sweetener, but they grow to a greater optical density, which is pretty cool. We're teaching E. coli to use a food it couldn't otherwise use in nature. Okay, that's enough science talk, but I had to get that in there. To end this presentation, I want to encourage you to think about areas of your life where you might apply a beginner's mindset. Maybe today it's answering a science fair judge's question with, I don't know. I would hypothesize, look, remember that I don't know is an acceptable answer in science. And for those of you heading to college soon, I think a beginner's mindset will be very helpful to you. Whether it's going to office hours with your professors and TAs, asking your friends and upperclassmen for help, or if you're like me, swallowing your pride and asking the RA how to use a washing machine, having the humility to ask questions will make your college experience better. I'll leave you with this. A good scientist isn't someone who always knows the answers and who never messes up. A good scientist is someone who is curious and humble enough to constantly ask questions and make mistakes. I hope you're all able to ask some good questions and make some good mistakes today. Thank you.
Hooray! Thank you, Daniel, so much for that amazing talk. Now we have a few minutes. If you have specific questions for Daniel about his talk, Beginner's Mindset, or anything else about his experience in research or his pathway to college, really the floor is kind of open. And while our participants think, Daniel, I would love to ask you a quick question about beginner's mindset. And so you said you stumbled upon the beginner's mindset and it changed your life. And I'm wondering, do you feel like you're always good at this or you're constantly working on it?
That's a great question. I absolutely am constantly working on that. I think probably many of us in this call feel the constant pull of perfectionism and the desire to project expertise. And that has continued for me even after I've gotten to college. So it's kind of a constant effort to remember to cut myself some slack and allow myself to approach problems like a beginner and not feel like I have to always have expertise, even though I am developing expertise, even though I am getting better at all of the research skills and procedures that I'm trying to learn about. I have to constantly remind myself to take a deep breath and apply that beginner's mindset.
Great. That's awesome. And I just have one more follow-up question. When you and I spoke, we've spoken a few times, but I think this may have been during your official alumni interview. You talked a lot about collaboration across interest and fields. I wonder if you can talk to our audience a little bit about your experience with that.
For sure. I think that's definitely a growing part of research, regardless of your field. The problems of the 21st century are so huge that they require teams across many disciplines to really make a dent in. And that's something I have been learning more and more through my research at NC State. So I'm housed in the chemical engineering department. We collaborate in departments across the university. For example, I do a lot of mathematical modeling as part of my research. So biological systems are so complex, we have to develop some pretty sophisticated models to understand exactly what's going on and predict what's going to happen. For example, with those viruses, I developed some models to predict exactly what proportion of the cells they're going to infect when we use them to transmit DNA. We also collaborate with food scientists, which was an unexpected kind of left turn for my research career. That wasn't something I was expecting. But for my other project, which involves using yeast to produce beta carotene to prevent vitamin A deficiency, we want to use those yeast in food cultures, but we're not food experts. So that requires a lot of collaboration with nutritionists and food scientists to understand what exactly is the best way to deliver these nutrients to the human body. So all of that to say, regardless of what your field is, if you continue in research, you will definitely have a lot of opportunities to interact across kind of scientific divides, because that is the only way really at this point to make an impact in the world, is to work interdisciplinarily.